actually good morning class of life science.
How
are you? I'm okay.
Yes.
Today we're going to introduce a new topic. But before we can introduce any new topic, I want us to go back where we were yesterday. We spoke about the cell function, cell structure and functions. So I want to know if you still remember those cell structures and the functions of the organs in the cell. No? So can anyone tell me anything about differences? Tell me the difference between animal cell and plant cell. Yes.
A plant cell has a cell membrane and a cell wall, whereas the animal cell has only the cell membrane.
Yes, that's true.
The plant cell has a large vacuole, and the animal cell has a small or no vacuoles.
That's true.
So, that's.
Anyone to help you? Anyone to continue with the information? You want to know more? What did you learn about the differences between animal cell and plant cell? What is it? What the difference? What did you pick up? What differences do you know? Do you still remember what you have learned yesterday? If yes, tell me the difference between plant cell and animal cell. Yes.
The plant cell has a cell wall and the animal cell has no cell wall.
Yes.
And the plant cell have large vacuole and animal cell have no vacuole whatsoever.
Yes, that's true. Any other thing? You want to say something? I want you to say something.
Plant cell has no shape.
Yes.
And animal cell has a shape.
And also the animal cell has a chloroplast, and also the plant cell has a chloroplast, and the animal cell does not have one.
Okay.
And also
Chloroplast. Plant cell has chloroplast and the animal cell does not have one.
yes.
Okay.
And also using a light microscope to view the cells of a plant and animal cells. Animal cells has a lysosome and a plant cell does not have a lysosome. And a plant and animal cell has a rough endoplasmic reticulum and a plant cell does not have a rough, it has smooth endoplasmic reticulum.
That's true. Okay. Can you tell me the functions of the cells, the functions of those organs? What are the organs of those cells? Tell me all the organs, like the organs in the animal cell and the organs in plant cell. Some of the organs I know they are similar and some of the organs they are not similar. Because the animal cell has this other part while the other one, a plant cell, does not have other organs. So I want to know the organs in plant cell and the organs in animal cell.
So, the plant cell has a cell wall, right? That gives shape to the cell.
Yes.
And whereas the plant cell does not have a cell wall. Yes.
Another organ from plant cell, animal cell.
Animal cell has a nucleus, even plant cell has a nucleus, and they also have a cell membrane and also cytoplasm and vacuoles. Sometimes an animal cell can have no vacuoles.
Okay. What's the function of nucleus?
The function of a nucleus, it controls all the activities inside the cells.
That's true. It controls all the activities inside the cells. Any other organ that you can talk about? There are lots of organs that you didn't mention. Yes?
Cell wall.
Cell wall.
It is responsible for the coming in of the cells coming out of the cell.
That's true. That's very true. Cell wall is responsible for the coming in of the cells and the going out of the cells.
Yes.
Whatever it enters and whatever it goes out, it's responsible for all those. Yes. Any other person?
The cytoplasm.
The cytoplasm.
The cytoplasm contains the cell of the cell.
That's true. It contains the cell set of the cell. Any other thing to add? Anything?
And also the cell membrane, it controls all the substances that comes in contact with the cells.
Yes. Let me check it as well. Okay. I can see that you have learned, even though some of you are not that confident in what you have learned. Or maybe you don't remember what they have learned. I don't know how come. But well, there is no problem. We're still gonna go through it again and again until it gets into your head. No?
Yes.
So now we are introducing a new concept of today. What we're going to learn today is the chapter 5. We're going to go on the chapter 5, support and the transport system in plant. But we are going to concentrate on the anatomy of dicotyledonous plant. That's what we are going to learn about. Is there anyone who knows anything about anatomy of dicotyledonous plant?
I don't know about the structure.
You know the structure?
Yes.
Okay.
So like the epidermis, right? The epidermis is made up of many layers. It's closely packed cells.
Yes.
Yes.
From India.
Yes. Epidermis is made up of closely packed cells.
And it helps the root from, it protects the root from injury.
Yes, and it protects the roots from injury. That's true. Today we are going to learn about the anatomy of dicotyledonous plant. The anatomy of a plant is its internal structure. You have already studied the internal structure of a dicotyledonous leaf. In this unit, you will learn about the structure and functions of tissue that makes up the roots and stem in dicotyledonous plant. Number 1, the root. The root consists of a number of different tissue types. The first is root structure. The different types of tissue in a dicotyledonous root from outside to inside are 1, epidermis, which is a layer of closely packed cell that protects the roots from injury. There is no waxy layer covering the cells. This allows water to be taken up by the root. Near the tip of the root, there is a root hair cell. Cortex it is made up of many layers of thin-walled parenchyma cells. The function of this cell is to transport water from the root hair to the xylem, as well as to store starch. The third one is endodermis, which is a single layer of cells found around the central vascular tissue. The cell wall of the endodermis have a waxy, waterproof band called the Casparian strip. This helps to channel water into the xylem. The fourth one is pericycle. The pericycle is formed by a single layer of parenchyma cells. Lateral root or slight roots are formed from this layer. Vascular tissue is the last one, which is the center of the root and is vascular tissue. It consists of xylem, phloem, cambium. Yes, it consists of xylem, phloem, and cambium. A cambium cell can divide by mitosis and produce more xylem and phloem cell. Let me give a brief about the stem and later the secondary thickening. So the stem has the epidermis, which I've told you what it means, the cortex, I've already spoke about it, and the third one is the vascular bundle, So the vascular bundle are arranged in a ring. The xylem is on the inner side and the phloem is on the outer side of the vascular bundle. The cambium separates the xylem from the phloem. Sclerenchyma tissue surrounds the vascular tissue, provides support for vascular bundle. The fourth one is the pith. The central pith is made up of parenchyma cells, and starch is stored in the cell. The third one is the secondary thickening. As I said, the first one is root, the second one is stem, and the third one is secondary thickening. So the process of secondary thickening in this vascular bundle of a young dicotyledonous stem, the xylem and the phloem are separated by vascular cambium. Vascular cambium produces new cells by mitosis or cell division. Before secondary thickening or growth starts, the vascular cambium links up to form a complete cambium ring in the stem of dicotyledonous plant. The cambium produces new xylem towards the inside and new phloem towards the outside of the stem. The new xylem and phloem are referred to as the secondary xylem and the secondary phloem. Every year, the cambium produces a new ring of secondary xylem and secondary phloem. The secondary xylem tissue consists of thick-walled vessel and forms wood. The active or new xylem tissue is closest to the cambium. The tissue transports water and mineral salts up the stem. The old xylem cell, filled with waste matter, are found nearer the center of the stem. The cambium and the phloem are pushed out to form a narrow layer known as the inner bark. The outermost part of the stem of tree or woody shrubs is called the outer bark. The outer bark is composed of special cells called cork, which is produced from the cork cambium. Cork cells are coated with an oily material called suberin or suberin. Actually, it's suberin, which protects the cells against water loss. Okay, now it's time to watch the movie or the video of the anatomy of the dicotyledonous plant. After watching of the video, you are now going to discuss amongst each other what you understand. I want you to learn from each other. Discussion is more of learning from each other, and it helps even with self-esteem. It improves the skills. The skills of what? Skills for what? For confidence, for you to talk to your peers. Yes. You must have confidence. That's a skill. So I want you. I'm going to give you a time after watching the video to discuss among yourself, and after discussion, then I'm going to ask you some few questions about what you have learned and so forth.
About anatomy of dicot root. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So when we talk about dicot root anatomy, we will find some layers. And the first layer is the outermost layer, that is called epidermis. And here you will see that some root hairs are found here. So basically, each cell of this epidermis can form the root hair. But all cells will not form root hairs, but some of them will form root hair. And each root hair represents a single cell. So it is the unicellular root hair. Next to epidermis, we can find this wide layer that is called cortex. So cortex contains several layers of cells, and do you know which type of cell is this? This is actually parenchyma cell, and here you can find many cells, many parenchymatous cells are there, and they have little intercellular spaces between the cells. Next layer is endodermis. So basically endodermis is the innermost layer of cortex, and here you can find a single layer of cells. This brown one is the endodermis. Only one single layer of cells is present here, and these are barrel-shaped cells, and they do not have any intercellular spaces between them. And these endodermal cells have a special deposition of a water impermeable waxy material that is called suberin in the form of Casparian strips. So endodermal cells have a special type of material that is called suberin, that is wax-like material. Next to the endodermis, we can find pericycle. So these yellow cells are pericycles. And the initiation of lateral roots and vascular cambium during the secondary growth takes place in these pericycles. Okay, and next to the pericycle, you can find vascular bundles. So vascular bundles, what are vascular bundles? Vascular bundles contain xylem and phloem. So 2 to 4 vascular bundles you can find. So in this picture, we can find 1, 2, 3, 4. 4 vascular bundles. And here we have phloem, xylem, phloem, xylem in this pattern. Now there are 2 types of xylem in this case, protoxylem and metaxylem. Protoxylem is the xylem that is formed fast during the tissue formation, and metaxylem is formed later during the tissue formation. And you can see that protoxylem, actually it lies towards the periphery. So you can see I am highlighting this spot. So protoxylem lies towards periphery and metaxylem lies towards the center. And this type of arrangement is called exarch. That means the protoxylem lies towards periphery and metaxylem is found towards the center. This type of special arrangement is called exarch. Then you can find conjunctive tissues. These green cells are conjunctive tissues. So you can find them between xylem and phloem, just like this. Next is peak. So peak is just the central part. It is the center, and in this dicot root, the peak is very small and inconspicuous. Inconspicuous means that it's not very much distinct, not very much clear. And all the tissues on the inner side of the endodermis, that means first is pericycle, then you can find vascular bundles, then you can find conjunctive tissue, and last the peak. So all of them will make the stem. So this is all about today's lecture. I think you understood the session.
Anatomy of a dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the following parts: epidermis, cortex, pericycle, vascular bundle, and pith. Epidermis. It is the outermost protective layer of the stem covered by cuticle. It may bear trichomes and a few stomata. Cortex. It is present between epidermis and pericycle. It is divided into 3 parts, namely hypodermis, cortical layer, and endodermis. It contains few layer of parenchymatous cells and parenchymatous cells with conspicuous intercellular spaces. Rich in starch. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as a starch sheath. Pericycle. On the inside of endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Vascular bundle. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. Vascular bundles are arranged in a ring. This arrangement is a characteristic of dicot stem. They are conjoined, open, and have enlarged protoxylem. Fifth, a large number of rounded parenchymatous cells with large intercellular spaces, which occupy the central portion of the stem, constitute the pith. Anatomy of a monocotyledonous stem. Epidermis, covered by cuticle. Cortex. The monocot stem has a sclerenchymatous hypodermis, rich in starch. Pericycle is same as dicot stem. Vascular bundles are scattered and closed, with peripheral bundles being smaller than central. Phloem. Parenchyma is absent and water-containing cavities are present. Pith is absent. Anatomy of a dorsiventral dicotyledonous leaf. A vertical section of a dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. Epidermis covers both upper adaxial, bearing less, no stomata, and lower abaxial, bearing more stomata, surface of leaf, and bears a cuticle. Mesophyll. The tissue between the upper and the lower epidermis is called the mesophyll. There are numerous large spaces and air cavities between these cells. Differentiated into palisade parenchyma, has parallel arranged elongated cells, and spongy parenchyma with loosely arranged oval round cells, which extends to the lower epidermis. It possesses chlorophyll and carries out photosynthesis. Vascular bundle. Present in midrib and veins, reticulate venation, surrounded by bundle-shaped cells. Anatomy of isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. Epidermis. Stomata present on both sides. Mesophyll. Not differentiated into palisade and spongy parenchyma. Vascular bundles. Leaves have a parallel venation. Bulliform cells, modified epidermal cells, are present along the vein. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize water loss. Of the roots and stems in length with the help of apical meristem is called the primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called the secondary growth. The tissues involved in secondary growth are the two lateral meristems. A. Vascular cambium, and B. Cork cambium. A. Vascular cambium. The meristematic layer that is responsible for cutting off vascular tissues, xylem and phloem, is called vascular cambium. In the young stem, it is present in patches as a single layer between the xylem and phloem. Later, it forms a complete ring. Formation formation of cambial ring. In dicot stems, the cells of cambium present between primary xylem and primary phloem is the interfascicular cambium. The cells of medullary cells adjoining these intrafascicular cambium become meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells, both towards the inner and the outer sides. The cells cut off towards pith mature into secondary xylem, and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. The primary and secondary phloems get gradually crushed due to the continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact in or around the center. At some places, the cambium forms a narrow band of parenchyma, which passes through the secondary xylem and the secondary phloem in the radial directions. These are the secondary medullary rays. Springwood and atomwood. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform through the year. In the spring season, cambium is very active and produces a large number of xylary elements, having vessels with wider cavities. The wood formed during this season is called springwood or early wood. In winter, the cambium is less active and forms fewer xylary elements that have narrow vessels, and this wood is called atomwood or late wood. The springwood is lighter in color and has a lower density, whereas the atomwood is darker and has higher density. The two kinds of woods that appear as alternate concentric rings constitute an annual ring. Annual rings seen in a cut stem give an estimate of the age of the tree. Heartwood and sapwood. In oak trees, the greater part of secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layers of the stem. These substances make it hard, durable, and resistant to the attacks of microorganisms and insects. This region comprises dead elements with highly lignified walls and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as the sapwood. It is involved in the conduction of water and minerals from root to leaf.
Guys, then I'm here. Yeah, in the Venus with Casparian scripts like Vishisha.
So the root consists of number of different the structure. They consider from the outside to the inside are the epidermis, cortex, endodermis, pericycle, and the so in that form I only studied three of them, which is the epidermis, the cortex, and the pericycle. So they are parenchyma cells. And lateral root, side root are formed from the side. So that is where I really understand the different types of tissues in the root. So yes, that's why I say it. You've got to train someone else so that you can train someone.
Structures are
Wound. Okay, you can check this part. So we are going to tell you about the function of the root. So the root enters the plant to the soil and helps it absorb water and dissolve minerals from the soil. Okay. Which means it is the function of the root to a plant. It helps the plant. Right? So we are practice how far I got about the root. Yes, let's go ahead and
It's like the thing I don't understand is that we must know like what is labeled there.
yes.
It protects the root from injury. Yes, and also protects it beneath epidemics.
So here we have to know how it is labeled, right? Know how it is labeled. and know how to divide the letters. Yes.
All we must know is the structure and functions of it.
The structure and functions of the root. Then we go to the next page, which we are going to start introducing about the stem.
Because we will need to know that.
Yes, we need to know that. Yes.
The names of the plant.
Are we going to find them all? That's the thing.
And now let's discuss it in the next slide.
So, and I think it's
But as long as you know where
I would want to ask you questions. I believe that you have learned a lot from the video, and I also believe that you have learned a lot from your discussion because you've watched the video and you did your discussions. So, tell me what you have learned in your discussion.
I learned about the vascular tissue, which is the center of the root, where it consists of a cannabis, a xylem, and a phloem, where a cannabis, the cells of the cannabis divides by mitosis to produce more xylem and phloem.
Okay. Another person? Yes. Julia?
I learned about the function of the roots, that it absorbs water and also dissolve minerals from the soil. And also I learned about the structure of the roots, which is a cortex that is made up of many layers which are very thin walled parenchyma cells, which its function is that it transports water from the roots hair to the extreme, and it also stores starch.
Okay. Clap hands for them all. Even those who didn't say much or didn't say anything now about what they have learned, because I believe that one person or two didn't say anything. It's one, eh? So I believe that with writing, they will say a lot. Okay, let me give you a chance and you write your own classwork. now that we are done writing our classwork, I want us to go and discuss the corrections.
Yes.
So the first question, list 2 ways a root can be identified from its internal structure. List 2 ways a root can be identified from its internal structure. What are those 2 ways?
Phloem and xylem.
Phloem and xylem, that's correct. That's correct. Okay, the second one. The other one is phloem and xylem occupying the central stele. And another one, what can you say? There are actually 4 ways, but she does listed 2 ways. Yes?
And I said, by the waxy surface of the epidermis that protected from injury.
No. Yes?
Columbia.
Columbium.
Yeah.
No.
Wow.
The pericycle. No. The second one, root's hair are present.
Yes.
These 2 ways the root can be identified from its internal structure. The root's hair are present. Then if you got it, then you are correct.
Yeah.
Then number 3, xylem alternate with phloem. Xylem alternate with phloem. You can see xylem there and phloem. They work together.
Yes.
They work hand in hand. You have to show that it is alternating with xylem. Okay, the third one, a clearly defined endodermis is present. The endodermis is present. Clearly defined endodermis is present. A clearly defined endodermis is present.
Yes.
Then 2.2, name 2 finger-like outgrowth of the epidermis of roots. Finger-like outgrowth of the epidermis of roots. What are those? Yes? Root hair, that's correct. Another one?
I wrote
No.
Casparian strips.
What?
Casparian strips.
No. Root hair? The second one? Pericycle, yes?
Is it epidemic?
No, it's a pericycle. The pericycle. These are the finger-like growth of the epidermis.
Oh, yes.
The finger-like. Okay, number 3, name 4 tissue that make up the stele of root. 4 tissues that make up the stele of the root. The 4 tissues that makes the central part of the root. Yes? Pericycle, that's correct. Phloem, that's correct. Xylem, that's correct. Another one? Cambia, that's correct. Another one? Other one can say pith.
The pith, yes.
The pith.
Yes.
Have you seen the pith on the diagram?
Yes.
Yes, that is a pith. Ne?
Yes,
But
ma'am.
if you got 4 of them, then you are on the right track.
Yes.
Can I have the light, please?
So thank you very much. This is our end of the lesson. So we are going to continue with the very same lesson tomorrow. We're going to learn more, we're going to discuss more, we're going to see more of videos, we're going to get more of information. So let's meet again tomorrow and we discuss the same thing. We speak